Okay, thank you everyone. So my name is Filippo Utro. I'm going to talk, can you hear me? Okay, my name is Filippo Utro. I'm going to talk on accelerated discovery in molecular biology. So, okay, as you all know, the development of a drug is a long and expensive journey. It might take over a decade, $1 million to put a drug on the market. In IBM research, we are working on creating new technology to accelerate this process. We develop a series of tools and release them as open source that can handle a variety of data from imaging, clinical record, histocartography, single cell, omics, and so on. So today, I'm going to focus on one specific of them called Genome4SD. So Genome4SD stands for Genomic Toolkit for Scientific Discovery. It takes a multi-mode and is able to handle multi-omics data and allow the analysis of patients in health and disease states over time or under specific treatments in order to deliver clinical action. You can use Genome4SD as a standalone tool, or you can use its outputs or its functionalities for downstream analysis, for instance, as a machine learning or an extra task you might have in your project. So just to give a bit of a background, how Genome4SD, why we need it, why it was born, we did a partnership with the Broad Institute in Boston, which was focused on understand and study drug resistance. So we collected a sample for different patients under different cancer type on the the treatment, during the treatment. We collect in some case multiple times during the treatment, then post the treatment to see and analyze when the resistance was developed and which was the rationale behind the resistance. So due to this particular kind of data, we then need new technologies, new approach. I'm going to introduce some of them today. Due to the limited time, I'm not going to go under the hood of each of the methods, but we can talk them if you want offline. I just want to give you just a quick idea behind it. What is the use? What is the rationale? And we already have some early results that have been published in the last few years. I'll post them, some of them. So the first method that I'm going to present is called lesion shedding model. So we collect the sample longitudinally, often through the blood, because as you know, cancer shed tumor cell in the blood that can be collected to ctDNA or cell-free DNA. The question now one can ask, how much each lesion is shedding? Is there a different lesion is shedding more than the other? Are we capturing completely the entire tumor or the different kind of tumor in all the lesion in the body? Is that working well? Or is that a way from the cfDNA figure out if the lesion is responding better than the other without any extra invasive approach? So the first question we asked is how much each lesion is shedding in the blood. And what we did, we developed this lesion shedding model. It was recently published on Griffin Bioinformatics. There is the reference of the paper if you're interested. So the idea is that you take as input the DNA profile, the molecular profile of the lesion and the one in the blood, and then you create a synthetic blood with different weights of shedding to the different lesion, and you create multiple this synthetic blood and see which one fits most closely to the real blood, and able to do a sort of ranking across the different lesions. So we are able to create this sort of direct network where each node here is a lesion, and they are color-coded based on the different location where the lesion is in the body. So in this case, we have multiple lesions from the liver and few from the lung, from the two different lungs of the patient. And we can see that some lesion is shedding more than the other one. In that case, if one lesion precedes another one during shedding, there is a direct edge between the one that's ranking more than the other one. And you can, if you want, simplify that view in this high level between high, medium, and low ranking shedding. We tested this and we analyzed this approach on a breast cancer patient, an ER-positive breast cancer patient. And in case of multiple blood sample, we can also track how much the lesion shedding is changing crossing over time. So we can see we've created this kind of subway of background plot, which each line is a different lesion, and with some confidence interval, how much is that lesion actually shedding and how it changes over time. What we see on the right is two different patients, and I want to take your attention on the two lesions that are highlighted in purple. which we were able to detect in the blood before they were actually being detected in imaging or any extra analysis. For instance, in the patient on the top, we can see that the top MRI that you see there was taken around 42 days. There is no lesion called lesion 22, and that what I already see as a top-ranking eye shedding lesion from very beginning. And we can see that that lesion then was actually visible at day 90. So we're actually able to detect in the blood the lesion way before was easy to detect. And we can see how that behaves. And we can see the similar picture on the other patient on the bottom. The X mark on the, if hopefully you can see, is where actually the lesion become detectable. Of course, when we work on time series data, clonal evolution and phylogeny is a very easy step approach that we want to analyze. So we want to track easily and more effectively the notion of time during this step. So we developed a tool called Concerti, which also helped to analyze and better visualize the time series impact of the data. And the framework is actually explained here. So it works here for the case of multiple lesions of the same body. So you have only one time point, let's say an autopsy when you have multiple lesions, or you have a serial longitudinal cfDNA sample or tumor biopsy across time. And the idea is behind this, basically, it's very simple. You start taking into account all alterations that are present in all your samples, cluster those, and then try to define, so which are the different clusters then going to be involved in the clone, and then consider the alterations that are unique to the singular sample or subset in of those, and then adding those to create all possible phylogeny, and then you score them and create which is the most realistic phylogeny. The penetrance of a clone and so on, for instance, you can use the VAFs or the variable allele frequencies, or if you want to have more sophisticated measure, you can use the cancer cell fraction, also CCF, which is also taking into account the purity of your sample or the number of ploidy, but it's a different measure. Clear example where Concerti or time series analysis can be used is leukemia, which is a blood cancer, a liquid cancer type, which is also easier sometimes to get sample through. It's a low mutation cancer, but it's also very impactful and very long disease, and that's because this is why it's also sometimes called chronic. This is why it's called chronic leukemia, right? So we're analyzing different treatments, BTK inhibitor, covalent and non-covalent, and we analyze how it's through Concerti validate this approach and see how easier to see possible resistance mechanism in the patients. So I'm going to give you a quick example on this patient. So this patient was a CLL patient under ibrutinib, and we can see on top of that, we have the visualization of the Concerti output through what is called a fish plot, that each color represents a cluster, and they are nested to each other to represent the biological clone, because each cluster is not, there is a meaning between the accumulate all the other alteration. The size of the fish plot is actually representing the tumor burden, and it's proxy of the tumor amount, or the tumor response through the white cell count. On the bottom, we have a more classical tree visualization, which they represent at each different time points of the dashed line, the different clones, and each cluster is nested to define the clone that are there. The sides of the node represent instead of the CCF, which is a different representation you can see from the tumor burden, because we have the white cell count that take into account. So we can see how the patient is initially responding to the treatment until the 12 months, where a new clone, the brown one and the green one, are born, creating that mechanism of resistance. Now you can see now, even in the fish plot, how it spikes up and how this clone takes over the cancer. And we can see that there are two known mechanisms of resistance in that clone, which are PLCG2 and BTK. Another example, slightly less pretty in this image, is another treatment for another patient, which is another BTK, non-covalent BTK inhibitor, which is pirtobrutinib, where we can see how the patient is performing. We can see the orange clone, which is actually take over, but then is being made responsible by Pirto. So Pirto is killing that clone by new mechanism of resistance. Another gatekeeper mutation in BTK is being born as a mechanism of resistance of Pirto. So we have the cancer is trying to escape through other series of mechanism of resistance. So trying to via concerti or so, it's possible to easily detect those, analyze those longitudinal data, and analyze and figure out which are those possible mechanisms of resistance. Another approach that I want to introduce is called instead REVEAL, which is instead the stochastic normalization approach for AI model to address the heterogeneity existing in the data. So REVEAL is basically performing a stochastic approach to try to use and extrapolate as much as possible all the information we have, even in non-genic region, which are often discarded by any of the analysis. So we always focus on just on the exonic and genomic on the transcription factor area, but we discard a large chunk of our genome, right, which is also called, without any particular annotation, which we refer here as the dark area of the genome, which is actually large part of what our genome is made. So we perform this analysis on try to classify 7 leukemia type on using all genome sample for around 700 patients, and using an off-the-shelf suite of machine learning, how much each of the different sections of your genome, so the exome, the genetic region, the annotated region, which include transcription factor binding site, and so on, a combination of those versus the dark area, and we can see that how the machine learning is poor performing on that. We can see in this sort of heat map or donut map, the F1 representation, and now the performance change when the REVEAL is applied for this stochastic normalization. We can see how the non-coding genome under different selector pressure per different cancer type perform throughout and we can see then going further and trying to figure out what's going on in this particular, why that is interesting, try to see which are the features that are driving those in this selected area. We can go and see, discover new area which can be relevant, for instance, to identify those areas in the genome, and we can see those. And this paper was recently published on PLOS Computational Biology. Another paper or work we've been doing on epidemiology is a tool called RubricoE, which was recently accepted on Recom Genetics and is going to appear in iScience, is basically a framework to capture stable set of features that maximize the contributed variability. It's very useful when you do GWAS analysis. So it's an SVM or machine learning pipeline for variability base to feature rank, every cross, validate a section of different feature sets. We apply RubricoE on different contexts. We apply in Alzheimer, COVID, under for data, large amount of data such as the UK Biobank performing novel statistical analysis on that. So I give you, of course, some of the example of some of the tools that are present in Genome4SD. But again, Genome4SD is always as open source, is still under development. So if you have any question or issue you encounter, or you see, oh, this feature is nice, we would like to introduce, we can work together, just contact me anytime, open an issue on GitHub. We are open to work on any of that. But now also, I want to slightly switch gear and show what we did in the recent past. I want to show what we're trying to do in the future, what is coming ahead of us, right? Of course, we want, as I said in the beginning, we want to accelerate drug discovery. And now we want to planning to do that. We have 2 paths that we are pursuing right now. One is through generative AI, so the use of foundation model. The other one, which is an early step, is through quantum technology. So the first one, the new model AI that we have been driven recently, is through the creation of foundation model. So on the left, we can see the classical paradigm we have been viewing so far. On the right, we have the one of foundation model, where drastically change the idea of integrating that we start from a large amount of data, which we start to creating a series of a gigantic foundation model, and then which we tune to tailor our specific application. So our idea is the one to create, develop a foundation model that captures the compound human body complex, and using a variety of data from clinical record, imaging, omics, and so on, to discover novel targets for an identification of novel target for drug discovery. Now, as I mentioned before, another step ahead is quantum technology, which is starting slowly to become available. You can even run a quantum simulator on your machine. There is a Qiskit toolkit which IBM developed, which is a Python interface. You can download, it's open source. There are video tutorials online. There is seminar series every Friday on YouTube and so on, which you can go and play around. It performs an off-the-shelf some of the quantum tool like QSVM, Q machine learning, and so on. You can go in there again and study the analysis. And what we are now want to do is like apply this quantum technology to life science and healthcare. So what we did, we tried then to focus on a specific aspect. In particular, we were trying to apply this quantum technology on single cell data. So we decided to use quantum machine learning, quantum topological data analysis on single cell, on a cell-centric view. We recently I believe last week, we actually post on arXiv our prospective paper on how quantum can help in this cell-centric view. It was a collaboration between different people around the world. And please feel free to post it, read it, give us our comments, and it'll be very appreciated. So in the last few minutes that I'm gonna have, I believe I have 4, I'm gonna give you like a quick example of what we're focused on, which can be an application for instance single cell. We know that genetic engineering of cells for immunotherapy is a tough task. Only 6 FDA-approved CAR T therapy has been done since 2019 because now you see what I was talking about. And we need to define and enhance T-cell therapy response on a specific motif. So there are, to expand the therapeutic scope and significant engineering, we want to, for instance, focus not just on cancer, but on solid tumor, or it was done mostly for liquid tumor. We want, for instance, expand on solid tumor, which is a different task, or neurodegenerative disease and so on. There is like a lot of opportunity for this therapy, but there is a series of main challenges when we know about CAR T, for instance, its toxicity, the precise target identification, the testing, and its cost, because it's quite very expensive, right? So recently, in IBM Research and UCSD published in December of the last year a paper on Science where they were focused on a library of 12 motifs and which developed over around 2,300 CAR T cells, but only 250 were possible to generate through experiments due to the cost, right? So what we did was to basically create a CNN to predict the toxicity on this 250 and then try to see. But again, 250 is not that large when you talk about classical machine learning, but they can be useful in quantum because there you use the entanglement and the ability to use the entire space to work on this thing. Another approach instead is more like foundation, which is called topological data analysis, which is a mathematical model to study and order interaction. And we want to use quantum in these technologies to help to solve some of the computational bottleneck. And I'm happy to take any question if you have.
One question because we are.
And of course, you can talk me offline anytime.
Then you can talk with him. No questions? Yeah, one. Okay.
Hi.
Hi.
So I was wondering if the lesion shedding model can be applied also to other diseases that are not related to cancer.
We didn't test it, but I believe so. Maybe we need some fine tuning, but yes, I believe it'll be possible. We can talk maybe offline and discuss the details if you want.
So very great talk. I was wondering how much generalizable are these methods for other type of drugs, such as antibody therapeutics, because you show BTKI inhibitor for cancer. Do you know if your methods can generalize?
Yes. I'm sure I just showed a portion. We did on CLL, but we tested this also on solid tumor, the shed on cell-free DNA. We tested on breast cancer, lung, and so on. I didn't report results, but yes,